हेलो स्टूडेंट्स, स्वागत है आपका सभी का एनसीईआरटी ऑडियो बुक में। आज हम क्लास 11 का चैप्टर 6 स्टार्ट करेंगे, एनाटॉमी ऑफ फ्लावरिंग प्लांट्स। You can very easily see the structural similarities and variations in the external morphology of the larger living organisms, both plants and animals. Similarly, if we were to study the internal structure, one also finds several similarities as well as differences. This chapter introduced you to the internal structure and functional organization of higher plants. Study of internal structure of plants is called anatomy. Plants have a cells as the basic unit. Cells are organized into tissues and in turns to the tissue are organized into organs. Different organs in the plants shows differences in their internal structure. Within angiosperms, the monocots and dicots are seen to be anatomically different. Internal structures also show the adaptations to diverse environments. 6.1 The tissue. The tissue is a group of the cells having a common origin and usually performing a common function. A plant is made up of different kinds of tissue. Tissues are classified into two main groups, namely meristematic and permanent tissues, based on whether the cells being formed are capable of dividing or not. 6.1.1 Meristematic tissue. Growth in the plants is largely restricted to the specialized region of the active cells division called meristem. Plants have different kinds of the meristems. The meristems which occurs at the tip of the roots and shoots and produce the primary tissues are called apical meristem. Root apical meristem occupies the tip of the root, while the shoot apical meristem occupies the distance with most regions of the stem's axis. During the formation of the leaves and the elongation of the stem, some cells left behind from the shoot apical meristem constitute the auxiliary bud. Such buds are present in the axial of the leaves and are capable of forming a branch or a flower. The meristem which occurs between the mature tissue is known as intercalary meristem. They occur in the grasses and regenerate the parts removed by the grazing animals, herbivorous. Both apical meristem and the intercalary meristems are primary meristem because they appear early in the life of the plants and contribute to the formation of primary plant body. The meristem that occurs in the mature region of the roots and shoot are of many plants, particularly those of the produce woody axis and proper, appear later the primary meristem called the secondary or lateral meristem. They are cylindrically meristem, fascicular vascular cambium, interfascicular cambium and cork cambium are the example of lateral meristem. These are responsible for producing the secondary tissue. Following divisions of the cells in the both primary and as well as secondary meristem, the newly formed cells becomes structurally and functionally specialized and lose the ability of divide. Such cells are termed the permanent or mature cells and constitute the permanent tissue. During the formation of the primary plant body, specific region of the apical meristem produce the dermis tissue, ground tissue and vascular tissues. Permanent tissues. The cells of the permanent tissue do not generally divide further. Permanent tissue having all cells similar in structure and functions are called simple tissues. Permanent tissues having many different types of the cells are called complex tissues. Simple tissues. A simple tissue is made up of only one type of the cells. The various simple tissues in the plants are parenchyma, collenchyma and sclerenchyma. Parenchyma forms the major component within the organs. The cells of the parenchyma are generally isodiametric. They may be spherical, oval, round, polygonal or elongated in shape. Their walls are thin and made up of cellulose. They may be either be closely packed or have a particular intercellular spaces. The parenchyma perform various functions like photosynthesis, storage and secretion. The collenchyma occurs in layers below the epidermis in most of the dicotyledonous plant. It is found either as the homologous layer of the patches It consists of the cells which are much thickened at the corner due to the deposition of the cellulose, hemicellulose and pectin. Collenchymatous cells may be oval, spherical and polygonal and often contain chloroplast. These cells assimilate food when they contain chloroplast. Intercellular spaces are absent. They provide mechanical support to the growing parts of the plant such as young stem and the petiole of the leaf. Scalenchyma consists of the long, narrow cells with thick and lignified cell wall having a few or numerous pits. They are usually dead and without protoplast. On the basis of variations in the form of structure, origin and development, scalenchymatous may be either fibers or sclerites. The fibers are the thick-walled, elongated and pointed cells, generally occurring in groups in various parts of the plants. The sclerites and the spherical ovals and cylindrical highly thickened dead cells with very narrow cavities lumen. These are commonly found in the fruit walls of the nuts, pulp of the fruit like guava, pear and saporota, seed coat of the legume and the leaves of the tea. Scalenchyma provides the mechanical support to the organs. Complex tissues. The complex tissues are made up of more than one type of the cells and these work together as a unit. Xylem and phloem constitute the complex tissues in plant. Xylem function as the conducting tissue for water and minerals from roots to the stem and leaves. It also provides mechanical strength to the plant parts. It is composed of four different kinds of the elements, namely tracheids, vessels, xylem fibers and xylem parenchyma. Gymnosperm lack vessels in their xylem. Tracheids are elongated or a tube-like cells with a thick lignified walls and tapering ends. These are dead and without the protoplasm. The inner layers of the cell wall is thickening which vary in form. In flowering plants, tracheids and vessels are the main water transporting elements. Vessels is the long cylindrical tube-like structure made up of many cells called vessels members, each with lignified walls and the large central cavity. The vessel cells are also devoid of protoplasm. Vessels membranes are interconnected through the perforation in their common walls. The presence of the vessels in the characteristic features of the angiosperm. Xylem fibers have a highly thickened walls and oblilateral central lumen. These may be either be separated or accepted. Xylem parenchyma cells are living or thin-walled and their cell wall are made up of cellulose. They store the food materials in the form of starch or fat and the other substance like a tannin. The radial conduction of the water takes place by the ray of parenchymatous cells. Primary xylem is of two types, protoxylem and metaxylem. The first formed primary xylem elements are called protoxylem and the latter formed primary xylem is called metaxylem. In stem, the protoxylem lies towards the central pith and the metaxylem lies towards the periphery of the region. This type of the primary xylem is called entarch. In roots, the protoxylem lies towards the periphery and metaxylem lies towards the center. Such arrangement of the primary xylem is called exarch. Phloem transport food material usually from leaves to the other parts of the plant. Phloem in angiosperm is composed of sieve tubes, elements, companion cells, phloem parenchyma, and phloem fibers. Gymnosperm have albuminous cells and sieve cells. They lack sieve tubes and companion cells. Sieve tubes elements are also long tube-like structure arranged longitudinally and are associated with the companion cells. Their end walls are perforated in the sieve-like manner to form a sieve plates. A mature sieve elements possess the peripheral cytoplasm and a large vacuole but lacks of the nucleus. The function of sieve tubes are controlled by the nucleus of companion cells. The companion cells are specialized parenchymatous cells which are closely associated with sieve tube elements. The sieve tube elements and the companion cells are connected by the pit fields present between their common longitudinal walls. The companion cells help in the maintaining the pressure gradient in the sieve tubes. Phloem parenchyma is made up of the elongated, tapering cylindrical cells which have a dense cytoplasm and nucleus. The cell wall is composed of the cellulose and has pits through which the plasmodesmatal connections exist between the cells. The phloem parenchyma stores food material and other substances like a resin, latex, and mucilage. Phloem parenchyma is absent in most of the monocotyledon. Phloem fibers are made up of sclerenchymatous cells. These are generally absent in the primary phloem, but are found in the secondary phloem. These are much elongated, branched, and have a pointed needle-like apex. The cell wall of the phloem fibers are quite thick. At maturity, these fibers lose their protoplasm and become dead. Phloem fibers of jute, flax, and hemp are used commercially. The first formed primary phloem consists of a narrow sieve tubes and is referred the protophlloem, and the latter formed the phloem has a bigger sieve tubes which is referred as the metaphloem. The tissue system. We were discussing types of the tissue based on the types of the cell present. Let us now consider how tissues vary depending on their location in the plant body. Their structure and function would also be dependent on the location. On the basis of their structure and location, there are three types of tissue system. These are the epidermal tissue system and the ground and the fundamental tissue system and the vascular and conducting tissue system. Epidermal tissue system. The epidermal tissue system forms a outermost covering of the whole plant body and comprise epidermal cells, stomata, and the epidermal appendages and the trichomes of hairs. The epidermis is the outermost layer of the primary plant body. It is made up of elongated and compactly arranged cells which form a continuous layer. Epidermis is usually single layered. Epidermal cell is the parenchymatous with a small amount of cytoplasm lining the cell wall and the large vacuole. The outside of the epidermis is often covered with the waxy thick layer called the cuticle, which prevents the loss of water. Cuticle is absent in the root. Stomata are structure present in the epidermis of the leaves. Stomata regulate the process of the transpiration and the gaseous exchange. Each stoma is composed of two bean-shaped cells known as guard cells, which enclose the stomatal pore. In grasses, the guard cells are dumbbell-shaped and the outer walls of the guard cells, away from the stomatal pore, are thin and inner walls towards the stomatal pores are highly thickened. The guard cells process chloroplast and regulate the opening and closing of stomata. Sometimes a few epidermal cells in the victini of the guard cells becomes specialized in their shape and size are known as subpseudoidally cells. The stomatal aperture, guard cells, and the surrounding subpseudoidally cells are together called the stomatal aperture. The cells of the epidermis bear a number of the hairs. The root hairs are unicellular elongations of the epidermis cells and help to absorb the water and minerals from the soil. On the stem of the epidermis, hairs is called trichomes. The trichomes in the shoot systems are usually multicellular. They may be branched or unbranched and soft and stiff. They may be even be secretory. The trichomes help in the preventing water loss due to the transpiration. The ground tissue system. All tissue except epidermis and the vascular bundle constitute the ground tissue. It consists of the simple tissues such as parenchyma, collenchyma, and sclerenchyma. Parenchymatous cells are usually present in the cortex and pericycle, pith, and medullary rays in the primary stem of the roots. In leaves, the ground tissue is consist of thin-walled chloroplast containing the cell and is called mesophyll. The vascular tissue system. The vascular system consists of the complex tissues, the phloem and the xylem. The xylem and phloem together constitute the vascular bundles. In dicotyledonous stem, cambium is present between the phloem and xylem. Such vascular bundles becomes the presence of the cambium possess the ability to form the secondary xylem and phloem tissue, and hence they are called the open vascular bundles. In the monocotyledonous plant, the vascular bundles have no cambium present in them, hence they do not form the secondary tissue. They are referred to as a closed. When the xylem and phloem within the vascular bundles are arranged in the alternate manner along with the different radii, arrangement is called the radial such as the roots. In co-joint type of the vascular bundles, the xylem and phloems are jointly situated along the same radius of vascular bundles. Such vascular bundles are common in stems and leaves. The cojoint and vascular bundles usually have two phloem located only on the outer side of the xylem. Anatomy of dicotyledonous and monocotyledonous plants. For a better understanding of the tissue organization of the roots, stems and the leaves, it is convenient to study the transverse section of the mature zones of these organs. Dicotyledonous root. Look at the figure, it shows the transverse section of the sunflower root. The internal tissue of the organization of the follow. The outermost layer is epilemma. Many of the cells of the epilemma protrude in the form of the unicellular root hairs. The cortex consists of the several layers of the thin-walled parenchymatous cells. With the intercellular spaces, the innermost layer of the cortex is called endodermis. It comprises the single layer of the barrel-shaped cells with the intercellular spaces. The tangential as well as the radial walls of the endodermal cells have a deposition of the water impermeable waxy material subrine in the form of capsarian strips. Next to the endodermis lies the few layers of the thick-walled parenchymatous cells referred to the pericycle. Initiation of the lateral roots and the vascular cambium during the secondary growth of the takes place in these cells. The pith and the small inconspicuous the parenchymatous cells which lies between the xylem and phloem are called conjunctive tissue. They are usually 2 or 4 xylem and phloem patches. Later the cambium ring develops between the xylem and phloem. All tissues on the inner side of the endodermis such as pericycle, vascular bundles and pith are constitute the stele. Monocotyledonous root. The anatomy of the monocotyledonous root is similar to the dicot in many respects. It has a epidermis, cortex, endodermis, pericycle, vascular bundle and pith. As compared to the dicot root which have fewer xylem bundles, there are usually more than the 6 xylem bundles in the monocot root. The pith is large and well developed. Monocotyledonous roots do not undergo the secondary growth. Dicotyledonous stem. The transverse section of the typical young dicotyledonous stem shows that the epidermis is the outermost protective layer of the stem, covered with a thin layer of the cuticle. It may bear trichomes and the few stomata. The cells arranged in the multiple layers between the epidermis and the pericycle constitute the cortex. It consists of 3 subzones. The outer hypodermis consists of a few layers of the parenchymatous cells just below the epidermis which provide mechanical strength to the young stem. Cortical layers below hypodermis consist of a rounded thin-walled parenchymatous cells with the conspicuous intercellular spaces. The innermost layer of the cortex is called the endodermis. The cells of the endodermis are rich in starch grains and the layer is also referred to as the starch sheath. Pericycle is present on the inner side of the endodermis and above the phloem in the form of semilunar patches of the sclerenchyma. In between the vascular bundles, there are a few layers of the radially placed parenchymatous cells, which constitute the medullary rays. A large number of the vascular bundles are arranged in a ring. The ring arranged of a vascular bundles is a characteristic of dicot stem. Each vascular bundle is cojoint, open and with the enlarged protoxylem. A large number of the rounded parenchymatous cells with a large intercellular spaces which occupy the central portion of the stem constitute the pith. Monocotyledonous stem. The monocot stem has the sclerenchymatous hypodermis, a large number of the scattered vascular bundles, each surrounded by the sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. Vascular bundles are cojoint and closed. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent and the water containing cavities are present within the vascular bundles. Dorsiventrally dicotyledonous leaf. A vertical section of the dorsiventral leaf through a lamina shows 3 main parts, namely epidermis, mesophyll and vascular system. The epidermis, which covers both the upper surface adaxial epidermis and lower surface abaxial epidermis of the leaf has a conspicuous cuticle. The abaxial epidermis generally bears more stomata than the adaxial epidermis. The latter may even lack stomata. The tissue between the upper and the lower epidermis is called the mesophyll. Mesophyll, which possess the chloroplast and carry out photosynthesis, is made up of the parenchyma. It has two types of the cells, pellucid parenchyma and spongy parenchyma. The adaxially placed pellucid parenchyma is made up of the elongated cell which are arranged vertically and parallel to each other. The oval or round or loosely arranged spongy parenchyma is situated below the pellucid cells which extend to the lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular system includes vascular bundles which may be seen in the veins and midrib. The size of the vascular bundles are dependent on the size of the veins. The veins vary in thickness in the reticulate venations of the dicot leaves. The vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. Look at figure and find the position of the xylem in the vascular bundle. Isobilateral monocotyledonous leaf. The anatomy of isobilateral leaf is similar to that the dorsiventral leaf in many ways. It shows the following characteristics difference in an isobilateral leaf. The stomata are present on both the surface of the epidermis, and the mesophyll is not differentiated into pellucid and spongy parenchyma. In grasses, certain adaxial epidermis cells along the vein modified themselves into large, empty, colorless cells. These are called bulliform cells. When the bulliform cells in the leaves have absorbed water are turgid, the leaf's surface is exposed. When they are flaccid due to water stress, they make a leaves curl inward to minimize the water loss. The parallel venation in monocot leaves is reflected in the near similar size of vascular bundles, except the main veins, as seen in the vertical section of these leaves. Secondary growth. The growth of the roots and the stem in the length with the help of the apical meristem is called the primary growth. Apart from the apical growth, most dicotyledonous plant exhibit an increase in girth. This increase is called the secondary growth. The tissues involved in the secondary growth are lateral meristem, vascular cambium and cork cambium. Vascular cambium. The meristematic layer that is responsible for the cutting of the vascular tissue, xylem and phloem, is called the vascular cambium, and the young stem it is present in patches as the single layer between the xylem and phloem. Later it forms the complete ring. Formation of cambium ring. In dicot stem, the cells of the cambium present in between the primary xylem and primary phloem is the interfascicular cambium. The cells of the medullary rays adjoint these interfascicular cambium becomes the meristematic and forms the interfascicular cambium. Thus, a continuous ring of the cambium is formed. Activity of the cambial ring. The cambial ring becomes active and begins to cut off new cells both towards the inner and outer side. The cells cut off towards the pith mature into secondary xylem, and the cells cut off towards the periphery mature into secondary phloem. The cambium is generally more active in the inner side than the outer side. As a result, amount of the secondary xylem produced is more than the secondary phloem and soon forms the compact mass. Primary and secondary phloems get gradually crushed due to the continuous formation and accumulation of the secondary xylem. The primary xylem, however, remains more or less intact in or around the center. At some places, the cambium forms a narrow band of the parenchyma, which passes through a secondary xylem and the secondary phloem in the radial direction. These are secondary medullary rays. Spring wood and autumn wood. The activity of the cambium is under the control many physiological and the environmental factors. In temperate zone, the climatic conditions are not uniform through the year. In the spring season, cambium is very active and produces a large number of the xylage elements having vessels with wide cavities. The wood formed during this season is called spring wood or early wood. In winter, the cambium is less active and forms fewer xylary elements that have narrow vessels, and this wood is called autumn wood or late wood. The spring wood is lighter in color and has a lower density, whereas autumn wood is darker and has a very higher density. The two kinds of the woods that appears as alternate concentric rings constitute as a annual ring. Annual rings seen in a cut stem give an estimate of the age of tree, heartwood and sapwood. In old trees, greater part of the secondary xylem is dark brown due to the deposition of organic compounds like tannins, resins, oils, gums, aromatic substance, and essential oils in the central and innermost layer of the stem. These substances make it hard, durable, and resistant to the attack of the microorganism and insect. This region comprises dead elements with highly lignified walls and is called heartwood. The heartwood does not conduct water, but it gives mechanical support to the stem. The peripheral region of the secondary xylem is lighter in color and known as the sapwood. It is involved in the conduction of the water and minerals from the root to leaf. Cork cambium. As the stem constitute to increase in the girth due to the activity of the vascular cambium, the outer cortical and epidermis layer get broken and need to replaced to provide the new protective layers. Hence, sooner or later, another meristematic tissue called cork cambium or paleozoon develops usually in the cortex region. Paleozoon is the couple of the layers thick. It is made up of narrow, thin, walled and nearly rectangular cells. Paleozoon cuts off on both sides. The outer cells differentiated into cork or phloem and the inner cells differentiate into secondary cortex and pallidum. The cork is impervious water due to submarine deposition in the cell wall. The cells of the secondary cortex and parenchymatous pallium and pallidum are collectively known as peridum. Due to the activity of the cork cambium, pressure builds up on the remaining layers, peripheral pelagian, and ultimately these layers die and slot off. Park is the non-technical term that refers to all tissues exterior to the vascular cambium. Therefore, including secondary phloem, bark refers to the number of the tissues types, which periderm and the secondary phloem. Bark that is formed early in the season is called early or soft bark. Towards the end of the season, late and hard bark is formed. Name of the various kinds of the cells layers which constitute the bark. At certain region, the phalangean cuts off the closely arranged parenchymatous cells on the outer side instead of the cork cells. These parenchymatous cells soon rupture and epidermis forming a lens-shaped opening called lenticles. Lenticles permits the exchange of gases between the outer atmosphere and the innermost internal tissue of the stem. These and internal tissue of the stem. These occurs in the most woody trees. Secondary growth in roots. In the dicot root, the vascular cambium is completely secondary and origin. It originates from the tissue located just below the phloem bundles, a portion of the pericycle tissue above the protoxylem forming a complete and continuous wavy ring, which later becomes circulate. Further events are similar to those already described above for the dicotyledonous stem. Secondary growth also occurs in stem and roots of the gymnosperm. However, secondary growth does not occur in monocotyledonous. Here the lesson is completed. Thank you everyone.